Hello, my name is Meaghan Rose, and I am a fourth-year PhD student studying under Dr. Woo-Suk Chang here at the University of Texas at Arlington. During my time here, I've had the privilege of working on a variety of different projects that involve the symbiotic relationships between beneficial bacteria and their host plants. One of these particular projects involves the isolation and characterization of nitrogen-fixing methylotrophic bacteria from Crotalaria sagittalis root nodules. When most people think of nitrogen fixation and the symbiotic relationship, they think of bradyrhizobium, which is used for crop inoculation of soybeans all around the world to facilitate the nutrient cycling and therefore reduce the dependency on synthetic nitrogenous fertilizers. But one thing they don't consider is the great importance of carbon. All forms of life here on Earth are carbon-based and depend on it. The organisms utilize carbon and break it down further and further until eventually it reduces all the way down to a non-usable form, referred to as a reduced carbon source, a single carbon molecule, with two great examples of this being methanol and methane, with methanol being a major byproduct of plants. However, it was discovered that there is a particular type of bacteria, a methylotrophic bacteria, that can utilize these reduced carbon sources, as well as fixed atmospheric nitrogen and induce nodule formation on its host plants. These were originally isolated from a Crotalaria species, and so this sparked the interest in searching for Texas native Crotalaria plants that might have a similar type bacteria with the same system going on that could then be utilized for Texas native farmers to use for their Crotalaria juncea crops. To identify and locate these organisms, a Crotalaria sagittalis population was found about an hour northwest of here, and the plants were identified as the correct Crotalaria sagittalis species. Nodules were observed, and the plants were taken back to the lab where the root systems were cleaned off, root nodules were removed, surface sterilized, suspended in a liquid media, crushed, and then spread out onto a solid media for the isolation and characterization of their bacterial symbionts. After the bacterial isolates were obtained, they underwent metabolic testing, specifically using reduced minimal media that had only methanol as the provided carbon source, selecting for bacteria that can utilize this reduced carbon source metabolically. In addition to this, each of the isolates were tentatively identified using a 16S rRNA sequencing system that used 8F and 1541 universal primers for this identification. There were three unique colony types that were obtained from these root nodules, type A, B, and C, shown here, with A showing an ability to inhibit the massive and rapid growth of type B. In addition to this, type B seemed to be the only one capable of utilizing methanol thus far, while other reduced carbon sources will be observed as well in the future. And based on the identification, three colony types were identified to be different organisms, with type C being bradyrhizobium, type A being Bacillus firmus or Cytobacillus, and type B being Paenibacillus. All three of these organisms are known plant symbionts, with bradyrhizobium being sort of the poster child for nodulation as well as nitrogen fixation. Now, both bradyrhizobium as well as the Bacillus firmus do show the ability to induce nodulation in soybeans, and so these may perhaps be used as an inoculum for Texas native farmers that might utilize this particular crop. And Paenibacillus is also a known plant symbiont that shows an ability for nitrogen fixation, phosphate solubilization, the production of plant growth promoting hormones, as well as help to combat any sort of phytopathogens or nematode infections, which is a pretty big problem for any sort of farmer, particularly in the South. Now, more work will be done for these particular isolates, specifically focusing on molecular analysis with targeted PCR, looking for any genes that are related to methylotrophy, nitrogen fixation, or nodulation. Thank you.